video, we are going to look about another important molecular technique, which is AGE, Agarose gel electrophoresis. In short, we call it as AGE. So, from the name itself, we can understand the meaning of the technique. Electrophoresis means separating molecules with the electric current. So, gel electrophoresis means we separate these molecules in a gel. And agarose means the gel is made up of agarose. So, agarose gel electrophoresis is this technique. So what is this technique? Is a technique used to separate DNA or RNA molecules based on their size using electric current. So using electric current, we separate this DNA RNA molecules based on their size. So some DNA will be larger, some DNA will be shorter. So based on their size, we will be separating this DNA and RNA molecule. Who discovered this technique is Arne Theselius. Oh, I think it's someone from the Doctor Strange. So in 31, actually, there is no definitive person who discovered this technique, but most of the articles cite this man. So I have mentioned him. And apart from this, Schwartz and Cantor in 1984, they also discovered a technique called PFGE. Okay, this is also related to age. And why this technique is done is DNA for there are many applications for age. Some things to mention are DNA fingerprinting, forensic studies, so evolutionary studies like how animals are related to each other. And lastly, medical diagnosis. Just check the principle and procedure of this technique. The principle of this technique, it is very simple. DNAs are negatively charged molecules. So they have a phosphate group which is negatively charged. So because of that, all the DNA molecules are negatively charged. So what happens is when we apply electric current, its DNA will be moving towards a positively charged electrode, which is anode. This negatively charged DNA, while applying electric current, it will be attracted towards the positive charge. So inside a gel, there will be a matrix-like appearance, like a net or a sieve vessel. So in sieve vessel, what will happen? Smaller things will be going inside and coming out, but larger molecules will be staying in the sieve. So same like that, this larger DNA molecules will be stuck inside this matrix net, and it will be moving very slowly. But the smaller molecules will move faster, and by this we will separate the larger DNA molecules from the smaller DNA molecules. This is the basic separation technique for this age. Coming to the important components of this agarose gel electrophoresis are around five things. Actually, more things are there, but basic five things are very important. So, first thing is sample DNA. This sample DNA has to be chopped using restriction enzymes so that it can be with different sizes. So, we can't be separating a whole long DNA molecule in this agarose gel electrophoresis. So, we have to chop it into different sizes so that we can separate them based on different of their length. And the second important thing is loading dye or tracking dye. So we will add a glycerol or fecal. This will help the density of the sample. So some DNAs will be very small, so it might not sink well. It will be floating in the sample. So to sink it in the well, there we will create wells in the agarose gel. That well, it has to sink, so should not be float and coming out. So that we will to increase the density of the sample, we will add a glycerol or fecal. Apart from that, we will add a tracking dye, which is bromophenol blue or xylene cyanol. Okay, so this will help to track where our DNA is. Like if we are running for around 10 to 15 minutes, how long our DNA has been traveled, we can identify with this tracking gel. Third important and the most important thing is agarose. So this is the one which will make this gel. So generally we make 1% to 2%. So like mostly 1% to 1.5 2% of agarose. So the pore size will be 100 nanometer to 500 nanometer. So if you increase the concentration of the agarose, the pore size will decrease because it will help to separate smaller DNA. If your small DNA sizes are very smaller, you can put concentration of the agarose more so that the small pore size will be smaller and the smaller DNA will be separated. If your DNA samples are lengths are larger, then you can use less concentration of agarose so that the pore size will be a little bigger and you can separate larger DNA molecules. Coming to the fourth important thing is TAE or TBE buffer. So buffer used to stabilize our process. So DNA DNA from a negative electrode to positive electrode, this buffers will be helpful. So TAE means tris acetate EDTA, and TBE means tris borate EDTA. So these are the important buffers. And last important thing is ethidium bromide, ETBR. It is a very carcinogenic agent, so we have to handle with it with caution. And it is an intercalating agent. So what happens is it will helps to visualize our DNA bands in UV trans illuminator. Our DNA bands will be in orange color. It will help to locate our DNA where it has been separated. So apart from all these things, we need a microwave oven to dissolve the agarose gel, and a gel electrophoresis tank with a comb, border, and electrode, and UV trans illuminator to visualize, and last gel dock. So identify our DNA bands, how it is being separated. So these are the important materials. So coming to the procedure of this technique, we can separate into 12 simple steps. So first is preparation of sample DNA with restriction enzyme. So with restriction enzyme, we cut the sample DNA into small pieces. Second thing is mix this sample DNA with the loading dye. So in the loading dye, we will be having glycerol plus bromophenol blue. So we know the function of both of this before in the materials required step itself. Third thing is prepare agarose gel. So based on our need, we will determine the concentration of agarose and prepare the gel and add ETBR. So ETBR is an intercalating agent. It will help for visualization. Fifth important thing is cast the gel in the tray. So there will be a gel casting tray. So we will pour this gel and we will make it solidify, and our gel will be formed. Sixth is place the gel in the tank well near the cathode. Wells should be placed near the cathode. So while placing the gel in so while placing the gel in the tank, we have to keep the well near the cathode, near the negative charge, because from the well only it will start moving. The DNA will start moving. Fill the tank with the buffer. So now we fill the tank with we pour our TAE or TBE buffer in the tank and insert electrode respectively. So red color electrode means it is a anode which is positively charged. Black color electrode means it is cathode negatively charged, and we apply around 80 volt to 150 volt for 1 hour. So approximate timing. So it will based on the separation, it will differ. Up to the dye reaches 80% of the gel bottom. So when the tracking dye reaches the 80% of the bottom of the gel, we have to turn it off, and we have to take the apparatus very carefully. We use it in a UV trans illuminator, so which helps to visualize the DNA band, and we can take a pic of this gel, and we can identify where our DNA bands are separated using a gel dock. So based on the principle, the smaller DNA will be separated in the bottom of the gel, and the basic procedure of this agarose gel electrophoresis. In this video, we discussed the agarose gel electrophoresis. It's what is that? Its principle, its applications, and its materials. What are the important components of the agarose gel electrophoresis, and the procedure, how it has been done. So that's all.